Ingolf, would you start by explaining what is pharmacogenetics?
Yes, the term pharmacogenetics was coined due to the observation that individuals respond differentially to drugs. We know that people sometimes respond, others do not. Some people suffer from side effects and others do not. The question is, why is it so? And one part of the explanation is in our genes, that drugs are metabolized in a differential way in different velocities, or also the effect of a drug can be modulated by our genes. So that's why, how can genes contribute to drug response? That is the term of pharmacogenetics. There's only one more distinction. We have to distinguish between somatic variants being important in the treatment of tumors in oncology, and hereditary variants, which are responsible for our metabolism of drugs and also the drug action as it is in total. So pharmacogenetics more or less contributes to the fields outside of oncology, in cardiovascular diseases and psychiatry and so on.
And as an add-on question, how long has the concept been there and the technology really available to be able to do the studies, to be able to do the testing?
The observation have been made already in the 50s and early 60s that drugs are excreted with a different velocity into the urine. Now, with the development of molecular biology, we could identify genetic variants in the genes which contribute to this phenomenon, and that happens in the 80s. And now we have modern techniques available that we can sequence genes in a very rapid way and can identify many different variants.
So with that in mind, here's a concept around for 60 years, technology for 30 years. And Matthias, would you say something about the current stage of clinical implementation of the technology?
So I think we have a lot of evidence now that for some drug metabolizing enzymes, also other drug targets, there is evidence that adverse drug reactions can be avoided as well, the response can be improved. And this was the idea that we should not only talk about science, that some different consortiums in the world, particularly in the US, it was stimulated, but also the Dutch people, they thought bring together this knowledge as a evidence-based knowledge and writing some guidelines. So if you have identified patients with a specific genotype, which is clearly shown that the genotype leads to a loss of function of the enzyme, and so the functionality of the enzyme is not there, this could be really a result in adverse drug reactions if we treat these patients with a specific drug. And this was now the idea of, in terms of writing these guidelines. So we have these evidence-based guidelines, and then the implementation is to use the guidelines and try to implement this in clinical practice. This is one way. Of course, this is difficult because we have different countries. It is probably much more difficult than in US. It depends also on different languages, different rules, different legal aspects in Europe, for example. And there is currently also initiatives ongoing in Europe to implement this information. Another aspect in terms of implementation comes from the idea that we are now in a really good position to do a next generation sequencing, and we have currently cheap technologies available to have the genome data. We can have the genome data of the single patient. And if you do a preemptive genotyping, which means you can genotype a patient and then you can add this information in an electronic medical record system, then if the patient will be admitted to a hospital and if by chance he gets a drug like an oncology drug or a drug used for cardiovascular diseases or something else, then you can check whether there is a link to a specific genetic variant which is linked to a functional alteration of this enzyme, and this may lead to a selection of more appropriate selection of the drug, maybe dose adjusted treatment of the patient. And this is the idea of electronic medical records and as the additional aspect of preemptive genotyping. So implementation currently is difficult, of course. This is in the beginning, but this is the right way, I think, so to use the currently available knowledge. And of course, what we should say, pharmacogenetics couldn't explain individuality for all these maybe thousands of drugs. But at the end, if we would only explain 3 or 5%, it's not too bad. And this can be the start. We should start with drugs and genetic information where we have really good evidence, and we have good examples like mercaptopurine or abacavir, or these fantastic data in terms from Munir Mohammed's group from London and in the UK. So that adverse drug reactions like hypersensitivity can be explained if you preemptively test for the genetic variant.
So the potential is either to guide more sophisticated choice of treatment for effects or for avoiding harm. Edoardo, would you comment on a disease example? Epilepsy is common, it can be serious. In what way could pharmacogenetics be helpful to patients with epilepsy?
Epilepsy is one of the diseases in which there is a renewed interest in pharmacogenetics. First, because there are several forms of epilepsy that are genetically based, but this is another aspect. Concerning pharmacogenetics, there are at least two important examples. Concerning genes coding for drug metabolizing enzymes, for example, there is an interest in phenytoin. It's possible genotyping subjects, patients, for a certain specific enzyme, CYP2C9, to identify patients who will reach very high plasma concentration of phenytoin. And so this is an example of test that can be useful, but in this case, epileptologists are already aware of the possibility to individualize drug dosage by checking plasma concentration of this drug. But on the other side, the real advance in pharmacogenetic of epilepsy is what Matthias said before, that now is possible, based on the first example of biological genetic marker, specific allele of the HLA system, is possible to predict carbamazepine-induced Stevens-Johnson syndrome, that is a severe condition, sometimes fatal. But this was identified in a specific subpopulation, in a national population. But there is evidence that other genetic marker, other biological marker, may be will be possible to find association with other serious adverse effects due to anti-epileptic drugs. So this is a promising area.
You're making a very important point there, that it's important to consider the population in question to make sure you've got the right test for the right potential either benefit or risk. With that in mind, Adrián, would you say something about the global health perspectives and what we know about variability on the pharmacogenetic level in different populations?
Well, I think that Edoardo is giving a very nice example. We're not based on the previous example given by Matthias. Based on the Munir Mohammed data and some other carbamazepine side effects are related to certain genes that are mutations that are more frequent in Asians. So that changed the paradigms of how to treat, how to use drugs across the world. We were using the same dosage recommendation, assuming the same risk for everyone in every place. Now we know if the frequency of certain allele, which is related with certain inter-individual variation, let's say side effect is much more frequent in a place, probably we need to do a different risk management plan in terms of pharmacovigilance. If we're talking about cytochrome P450s or other drug metabolizing enzymes, and we know that poor metabolizer or ultra-rapids are more or less frequent in certain groups, probably we need just to keep in mind at population level, for a given subject, you never know. But as a public healthcare perspective, you need to recommend certain dosage. For instance, in certain population with extremely high risk levels of ultra-rapid metabolizers, probably the dosage should be adapted to those populations. This is perspective. If you ask me how important is that, the data that we have for the evaluation at the EMA Pharmacogenomics Working Party, now in Europe 32% of the drugs that have been evaluated with the central procedure has a biomarker included in the recommendation. So it's an emerging field, and the population perspective I think should be taken into account.
And from your own work in Latin America, what key messages have you seen there?
So Hispanics is the interesting thing in Hispanics is it is a population that's been mixed in the last 400 years. In Europe, so it was a melting pot. But there were some Indians, Amerindians with an Oriental background. So as you need to consider as a much more Oriental is a population, more Asian is. This is very interesting correlated with the carbamazepine data, for instance. You know, this is a drug very much used. As much poor is a population, so economic is related to that, more Amerindian it is. As much more Amerindian it is, much more Oriental relation have. So then some black slaves were taken from Africa and some Hispanics, but the Hispanics were very special because in my opinion, in our data, are Mediterranean Hispanics, probably those that were not adapted to new religion rules in the Middle Ages. So Jews mostly, but Jewish and those people related to this part of the Mediterranean have very high frequency of ultra rapids. So 400 years back, we make a mixture with the three ethnic groups, which are Orientals, Hispanics, later on some other people coming from the Middle East, and blacks. So then it depends. Of course, if you go to a big city with a lot of people, mixture is not relevant. But if you go to certain places in Mexico that have 200 million people and several thousand which are pure Amerindians, probably you cannot bring the same dosage and the same risk management plan than if you are using these drugs in a very rich area with a lot of white people. So we found extremely large differences with ultra rapid metabolism of CYP26. Among Indians, there are very few. But when we go to certain areas, for instance in Argentina, we found a colony of Jews with more than 18% of ultra rapids. So then this is, in terms of public health, of course, for a given subject, again, I don't think it's relevant.
Marja-Liisa, from Sweden, would you tell me something about the experience there of introducing tests into clinical practice? What are the common tests that are now being made available, the common patient groups which are benefiting?
Yes, we have been doing pharmacogenetic testing for the healthcare for about 20 years, starting quite early for just a few enzymes involved in the metabolism of mainly psychiatric drugs. And slowly this has been emerging, so there are now a larger number of genes that we are genotyping for today. Still most of if we discuss the cytochrome P450 and drug metabolism, it's really, you have to realize and keep in mind that genotyping, it's always categorizing individuals just to a few categories. And there is a large variability in the enzyme activity, for example, between the different genotype or predicted phenotype groups. So to predict for an individual patient what would be the metabolic capacity, what would be the dose required, is quite a big challenge. And we really have to take into consideration all the other sources of inter-individual variability, apart from genetics. Age can be important in certain cases. Drug-drug interactions are very important very often, especially in psychiatry, where most of the people actually are treated with many compounds at the same time, and they can influence each other's metabolism.
There are now two major issues of implementation. One, we have the issue of a regulation. What are the criteria of implementing pharmacogenetics into clinical practice? And the other more burning issue is the system of reimbursement. So these are the practical issues we are now facing, because from a practical point of view it is easy currently. So that's not the problem. But what are the criteria, for instance, in Sweden? When we look for Sweden, are there any rules now to consider pharmacogenetics in practice? Or what are the levels of evidence which are necessary to consider a trait as a useful marker?
Any department in a hospital or university hospital is free in a way to start doing if one feels confident that you have the evidence. And of course you have to show the evidence. It is scrutinized by the clinicians who are then ordering these tests, of course. But here are quite big differences between different European countries also.
And how's the situation in Italy?
The situation is quite similar, but we use much less than they use this test. As I said before, only in oncology, I mean, pharmacogenetics has entered in the system, but in the other fields not so much. But one problem that I see in Italy, but I suppose everywhere, is the diffusion of this private group that start to promise genotyping for everything, giving the magic bullet, the drug that can solve all the problem. And now, in this period in which every person can go to internet and look at these things, the problem is just to give a scientific validity of things, because this can be a risk. The promise of solving all the problem with tests done in private lab whose quality is not known, and then with this promise of finding the right drug for a specific disease.
I want to stress the ethics words because this is the main problem in Spain. We have the same situation like in Sweden. Anyone can develop anything in every public hospital, but there is a heavy promotion from the private sector, mostly the private clinics. So, and you find this type of promises. If you pay 800 euros and you buy my test, I will tell you exactly which drug. And these data are coming back to the public system. Sometimes patients are bringing those data with high expectations that clinicians cannot fulfill. So this this heavy promotion is creating an expectation that should be considered, I think. So at least a related problem with that, it is that we need to harmonize the lab test, because in different countries in Europe, in different hospitals, we are analyzing different things and we are giving the same name or the same definition for things that are coming from different data. So we need to define which is a poor metabolizer, which is an ultra-rapid, with genes, etc., etc. That is another barrier that we need to solve across Europe.
Currently it's more or less in a research level, but what we all observe is that the genes contribute only to some part of the story. And we are, of course, thinking about what contributes out to the static biomarker genetic variant. And what we learned, of course, is that genes are regulated by epigenetic factors, and that's a very interesting research issue currently. To make it available as a therapeutic biomarker, then we are far away. Perhaps in oncology, in some traits we can explain, and Matthias, you are involved in such research also in kidney cancer, and made some very interesting observations to how epigenetics can change, for instance, a drug transporter.
Right. Of course, I believe we should consider different levels, epigenetics, non-genetic factors, as mentioned by Marja-Liisa, and also to genetics. What I really believe for this idea in terms of implementation is openly saying we couldn't say that only a genetic variant will be the predominant leader for all the drugs in terms of improvement of therapy or avoidance of drug toxicity. So what we need, I'm completely convinced, we need algorithms which include more information on the epigenetic level, of the non-genetic level. Of course, most people don't talk about age or sex, and this is a very important confounding factor for this expression of function of enzymes or transporters, as well as the genetic information. And there is an example available, the warfarin story. Of course, there is an algorithm available. I mean, also people know that is non-genetic factors included, genetic factors included, drug interactions, amiodarone. So that is what we need in the future. And this is probably a little bit more difficult than only to stress the idea to use a single gene and 2 or 3 variants and test these patients before drugs will be prescribed. And if we honestly work in this field in the future, in the way of personalized medicine, stratified medicine, I really believe we need strong research, and we need these new technologies, and we need the idea that an algorithm should be used. And in principle, this is done in clinical medicine. If you treat a patient with an infectious disease or you treat a patient with heart failure, this is an algorithm. You don't look only on some clinical symptoms. You bring together symptoms with laboratory data and clinical symptoms. And this is what we need also for drug therapy.
Thank you all very much.
Thanks to you.